Well, thanks very much for this opportunity to be here. So I'm a trained molecular geneticist, and I've done genome work for many years. And I would consider myself more of a very enthusiastic user of the genome editing technologies. So I'm not going to claim to be an expert, and I'm not going to focus my talk on the genome editing technologies themselves. There are plenty of other people in the room who will be doing that. So what I've been asked to do, though, is to describe the genome research landscape, the types of experiments that we're supporting in genome science that are likely to be impacted by these very, very powerful new and emerging genome editing technologies. So that's what I'm going to do. I'm going to try to give you that landscape. And hopefully what this will do is help start the juices going to figure out how can we apply these genome editing tools to some critically important and challenging problems in environmental health science. So let me start off with just a little bit of historical perspective on genome research. So back in the, I'll call it the pre-genome era, actually back in the days when I first started my career, when we talked about genome science, it was for the most part talking about comparing genomes between different species. So it was a genome of a species. You were working in mice, you were working with humans, rats, flies, zebrafish. And for the most part, if you were good at what you were doing, you tried to do an inbred strain or a defined genetic background. And that's because it actually made the experiments a lot easier. So you could control for, say, litter sizes, behaviors, body weight, body composition, longevity. So again, it just made the experiments easier to predict outcomes. And furthermore, if you're using a defined genetic background, it promoted reproducibilities between laboratories. So if you're using, say, C57 Black 6J in Boston, you were likely to get the same types of results if you were in Atlanta or in other places across the United States. So for the most part, any phenotypic differences, if you were using an inbred strain in mice, and there were differences with an inbred strain in rat, many people attributed this to species-specific differences. Or if you found something in a mouse that you didn't find in humans, then the mouse must not be working. Okay, I wish I had a nickel for every time that someone actually used that terminology. And for the most part, there was no consideration about the impact of genetic background, about all of the sequence variations, actually we heard a little bit about this earlier today, that can be contributing to a toxicological trait. Well, that's the historical perspective. But now, with access to genome sequences and the very powerful genomics tools, we're in a very different place. In fact, I think in addition to doing, say, species-specific genome experiments looking at toxicity effects, and we would still do that in the National Toxicology Program where we have a defined genetic background. It's an F1 between C3 and B6. Now what we're really interested in doing is expanding this and using genome research to begin to understand mechanisms of toxicity and better understanding the genes and the pathways that are implicated in how an organism responds to environmental perturbations. So what I'm going to do over the next few minutes is select, well, I've put it in 4 different categories of genome research that it's related to, say, mechanisms of toxicity. So the first general area of this landscape, I'm calling experiments to take advantage of inherent, naturally occurring genetic variability within extant strains of mice and rats and other organisms. And we're not talking about humans today, but this could also apply to a whole variety of different experiments that are currently being done in humans. So the second general category that I'll talk about, and this is kind of the no-brainer category, since many of us are using the genome editing tools in this category. This is where I'll talk about the creation of, I'll say, synthetic genomes, things that don't naturally occur in the environment, but we use our tools to create these, specifically to test hypotheses about mechanisms of toxicity. The third general category of genome research is in this whole area of epigenetic science. I mean, we now recognize that there are multiple ways of changing how genes are being expressed and how genes work within an organism. And NIEHS is very interested in better understanding epigenetic mechanisms in response to environmental exposures. And the last category is a category that I've become increasingly interested in recent years, is in the second eukaryotic gene, or second genome within eukaryotic cells, and that's the mitochondrial genome. So we'll talk just a little bit about that, and hopefully what I can do is present some of the challenges, and hopefully some of the technology experts in the audience will have some quick answers for us. So let me start off with this whole area of focus around genetic variability. So it was in the late 1990s that actually Ken Pagan, who was the director of the Jackson Laboratory at the time, brought the mouse genetics community together and asked a very simple question. We have hundreds of different inbred strains of mice. How much phenotypic variability and how much genotypic variability exists within these strains? Now remember, in the late 1990s, we didn't have genome sequencing tools. We couldn't just go in and sequence the genome or search a database and extract all that information. So what the community decided to do is to go in and to phenotype a large panel of these mice, not just one strain or two strains, which was typically done in a number of laboratories, but let's take a panel of 40 different strains. And the community actually identified these strains as likely to have the greatest degree of genetic heterogeneity. And the question was, well, how much phenotypic variability is there in this panel of different mice? They're all mice, but they're different genetic backgrounds. And I think the result was just absolutely astonishing. And this is just one illustration. So one of the first questions that investigators at NIEHS and UNC and other places collaborated is to look at toxicity of a common drug that's on the market, acetaminophen. And what they found is that there is remarkable variability in different inbred strains to acetaminophen toxicity. There are some strains here that for a given dose exhibited a very acute and a very strong toxicity response. And there are other strains that could basically use acetaminophen as a food source. Excuse me. So they didn't really care. So it was this remarkable heterogeneity in response. And this is also true for a number of toxicants and environmental chemicals that NIEHS was interested in. This is just an example that was done by the National Toxicology Program, looking at the rate of benzene clearance in different inbred strains of mice. And you can see there's a tenfold difference in response. So what these experiments did was really highlighted that if we're doing experiments in mice with inbred strains, and we're trying to predict results in humans, there are no inbred strains of humans. Maybe there are identical twins, which is the closest thing you're going to have to an inbred strain. But so it really told us that we have to be a little smarter about how we're doing these types of toxicity assays. And that's another whole lecture. I'm not going to get into that this morning. I think that was actually part of the series of, or the workshop you had here on strain variability. But one of the most important things here that these experiments brought to our attention is that we wanted to know, well, what are the genes in these different strains of mice that are responsible for these differential effects? So which genes have sequence variations in them that are likely to contribute to making some animals essentially completely resistant to the acetaminophen toxicity, and other strains of mice with genes and variants in those genes that would contribute to sensitivity to acetaminophen. And if we could identify those genes, then that really gives us an insight into what potentially is the molecular mechanism of toxicity. So there was a big focus on this at NIEHS. In fact, NIEHS was the first institute at the NIH that actually put money forward to develop a SNP panel. And the SNP panel was used to actually map the genomes of these different mice. And so we discovered that that actually brought us a little closer to identifying which chromosomes had gene variants that contribute to these differential toxicities, but we needed more than that. And in fact, what we needed was a whole different set of tools of mice that had more genetic heterogeneity. Now, I don't have time in my 15 minutes or so to go through all the reasons why this is true. If you're interested, we can meet at the bar later on. I would be happy to take you through all of the details of this. But suffice it to say that the mouse genetics community and the rat genetics community and the Drosophila genetics community have actually risen to the challenge, and they've created these different panels. These are really population models of mice. And these are called the diversity outcross and the collaborative cross. And again, I don't want to go into all the details, but basically here, you're doing kind of a human-like experiment. You're not using a single inbred strain of mouse, but you're using now populations of mice that are genetically different. But they're genetically different in very defined ways. And again, we can talk about that later. But I called it controlled complexity, which makes it a lot easier to find the loci or the regions in the chromosomes that harbor the variants of genes that are responsible for differential toxicity. So we had access to these different tools. And at the same time, one of our grantees, Ted Choi and his colleagues at Predictive Biology, and actually a number of other investigators in the mouse genetics community thought, gee, wouldn't it be nice to be able to do quicker experiments? You know, mouse experiments using the diversity outcross and collaborative cross are nice, but they take time. If we could do these genetic experiments in vitro and looking at, say, in vitro toxicity, that would be great. So what Ted did, as using one of our SBIR funding mechanisms, is actually took a population of those mice, those diversity outcross mice or collaborative cross mice, and he made embryonic stem cells from each mouse within that line. So you now have a population of embryonic stem cells. So in each well of this microtiter dish, there are embryonic stem cells that have the genome structure from an individual mouse from within one of the populations of either diversity outcross or collaborative cross. So and the beauty of using embryonic stem cells is that you could do basic toxicity. So you put in a different chemical at different doses, and you look to see at what dose do the cells actually die. But if you're interested in, say, cardiomyocytes, you can take the embryonic stem cells and you can differentiate them into cardiomyocytes and look for cardiomyocyte toxicity or neurons. So very powerful approaches. And in both cases now you can do toxicity experiments, and using the powerful genetic tools that are inherent in these different lines of mice, you can map the loci that contain the genes with sequence variations. And this is a typical example of what you're left with. So again, there are a lot of details in this, but this is a region of a chromosome. There are a whole bunch of genes in this region of the chromosome. And this is one of our challenges. So we typically can map high resolution, find many different what we call candidate genes, but one of the challenges is to figure out, well, which of these genes in here is actually causative for influencing that toxicity between different strains. So there are multiple different genes. So right now we have to go through and take a single gene, do a homologous recombination. It's a big effort. But if the genome editing tools could come in with your promise for quickness, if you could quickly go through and change the sequence of many of these genes from one strain to another, it would allow us to very quickly get at which gene is actually responsible for toxicity. So that's the challenge. So from a complex trait perspective, this is true not only for the inbred strains, but this is true for lots of human experiments as well. You get down to this critical interval and you want to know which variation within which gene is associated with your phenotype. So let me move on to the second major category. So here's the challenge for the community. So figure out how we can do these experiments a lot faster so we can identify those genes with sequence variants that are associated with the differential toxicity. But in the second general category, I'm calling it creation of synthetic genomes. So this is where currently we're doing gene knockouts. So we can do conditional knockouts. We can turn off a gene at a specific time in a specific cell type. We're also very interested in creating single nucleotide variations in promoter regions, coding or non-coding regions. In some instances, we'd like to be able to create translocations and deletions. And we can actually do this with the homologous recombination tools that are available, but they're painfully slow and horrendously expensive. And most importantly, many of our grantees and people at NIEHS are using the genome editing tools to actually make mutations in genes in cell culture or in organisms that have previously been refractory to creating mutations at specific sites in the genomes. Doing mutagenesis in rats or in zebrafish and other organisms is now possible using these CRISPR applications. So these are the types of things that we're doing. And more recently, I've been absolutely fascinated by some of the approaches using the genome editing tools where you can create libraries of guide RNA, and you can be targeting individual genes in populations of cells, looking for mechanisms of toxicity. I think we're going to hear a lot more about this in either this afternoon or tomorrow morning. So very enthusiastic about this. But my challenge to the community is, so we'd like to be able to do these things faster and more efficiently. So what is it you can do for us? And I think for the most part, this is the category that many of us work in. And I know I personally have benefited from the genome editing tools. It's just truly remarkable how fast you can make a knockout of a gene compared to the old days when I used to do it using homologous recombination. Truly remarkable. So that's part of the challenge here. So let's go on now to the third category. And this third category is about epigenetics. We heard a little bit about this morning. But I always like to show this slide, partly because it's actually work from my own laboratory, but I think it also, this is kind of, I call this epigenetics in action. So you're looking at a number of mice on this slide, and typically if you're a geneticist, you think about, well, you've got some mice that are yellow and fat compared to a mouse that has a normal agouti coat color. Some of the mice are mottled to different degrees. So you would just assume that these mice carry different mutations. Some of them would confer the propensity to become yellow and fat, and others would confer this propensity to develop this mottled coat color. Okay, but the reality in this case is that these mice are genetically identical. It's an inbred strain of mouse, and they all carry a mutation called viable yellow, which in its highest penetrance form makes mice yellow and massively obese. So this is an 85-gram mouse compared to a 25-gram littermate. Again, they are genetically identical. What we demonstrated over 20 years ago is that there's a modification, there's a retroviral-like element that basically hijacks the normal expression of the agouti gene, okay, and it causes this phenotype. But the big question was, well, how is it that you have mice that carry this mutation but they are phenotypically normal, or mice that have partial expressivity of this gene? We actually call it a dominance hierarchy, or penetrance hierarchy. So normal mice all the way to the fully penetrant mice. Well, we went on to demonstrate that this is caused by this variable penetrance is caused by an epigenetic modification of the promoter. So normally this promoter drives the ectopic expression, but in the cases of mice that are phenotypically normal, that promoter has been epigenetically silenced. And in the case of model mice, some of the cells have the promoter that's epigenetically silenced. In other cases, it's not. Now, one of the really interesting features about this mutation that makes it still of great interest to NIEHS is that this penetrance hierarchy can be influenced by what you feed the mice or what you treat the mice with. And actually, Dana Dolinoy in the audience, where's Dana? I just saw you, there you are, has done a lot of work together with Randy Gerl's lab and really demonstrated that you can influence this penetrance hierarchy by feeding the mice, actually feeding the mothers of the embryos, different chemical compounds, bisphenol A, vitamin B12. So what they demonstrated is that you can shift this penetrance hierarchy, which means that you're actually shifting the epigenetic mark as a consequence of an environmental exposure. We've done a lot of work together with Randy Gerl's lab and really demonstrated. Well, that's interesting. Wow. Okay, good job. Hierarchy by feeding the mice, actually feeding the mothers of the embryos. Boy, is that what I saw? Different chemical compounds, bisphenol A. Okay, tell you what. Vitamin B12. Turn the timer off. Okay. So what they demonstrated is that you can shift this penetrance hierarchy. Speaking of shifting, which means that you're actually shifting the epigenetic mark as a consequence of an environmental. We've done a lot of work together with Randy Gerl's lab and really demonstrated. Well, that's interesting. Let's see. Could I speak over myself? That's a new one. Hierarchy by feeding the mice. Does anyone have any idea what's going on? The embryos. Boy, is that what I saw? Different chemical compounds, bisphenol A. Okay, vitamin B12. Turn the timer off. Okay, so what they demonstrated is that you can shift this penetrance hierarchy, which means that you're actually shifting the epigenetic mark
as a consequence of an environmental.
Okay. Well, this is a very interesting way of saying maybe you said too much. Time to end the presentation.
Speak over myself. That's a new one.
Right, exactly.
I don't have any idea what's going on.
Talking about having an impact. If you didn't hear it the first time. Okay, great. So now let's see. Do we have slides back? Oh, my timer just went to 0. Okay, let's go to almost the last slide. I just have a couple more, just one more general area to talk about. And so let's go. So this is where we were. Next slide. Okay. And so I think the challenge is, and we're going to hear more about this over the course of the next day and a half. The challenge is how to use these powerful genome editing tools to test hypotheses. In many instances, especially in human populations, we find a SNP, or there's a CPG that's methylated that's associated with smoking in mothers. Is that a causative SNP, or is it just a bystander SNP? So now, with these genome editing tools, you can actually go in and change the nucleotide and test directly whether that's causative. So in the last general category, I'm going to talk about the second genome within eukaryotic cells, and that's the mitochondrial genome. So for the most part, what I've discovered is that you have the mitochondrial biologists and the mitochondrial geneticists, the Doug Wallaces and others who study the mitochondrial genome and hope to convince you that every human illness is associated with something related to mitochondria. It's actually pretty compelling. But then you also have a lot of other people like myself who study the nuclear genome. And never the two really mix that much, which is unfortunate because in the cell, there's a whole bunch of crosstalk that's actually happening. And this slide, what I'm illustrating is that these are a bunch of reactions that are associated with, say, epigenetic modifications that happen in the nucleus. But there are metabolites generated in the mitochondria. And just in the limitation of time, I want to focus on alpha-ketoglutarate. Remember the Krebs cycle? You thought you'd never have to remember some of the details on that. It's coming back to haunt you again. And acetyl-CoA, and all of these metabolites. These are generated in the mitochondria. These are either cofactors for the enzymes in the nucleus that can influence the epigenome, or they're substrates for example, acetylation of histones. So what we're testing right now is, are these metabolites, and is mitochondrial function directly connected with changing gene expression in the nucleus through epigenetic mechanisms? So why is this important? Why is it of interest to NIEHS? Well, the reality is that a lot of environmental toxicants have their deleterious effects on the organism by hitting the mitochondria. So if we better understand how the mitochondria communicates with the nucleus, we'll have a better sense of understanding how those mitochondrial toxicants are actually influencing the biology of the cell and the biology of the organism. So I think that's really it. So the challenge is really to figure out, can the genome editing tools that are targeting different genes across the nuclear genome, what can we do about making specific mutations in genes in the mitochondria? The limited data that we have thus far is that the type of impact on the epigenome, if you completely delete the mitochondrial DNA, is different than if you, in fact, it's opposite to what you see on the epigenetic effects on the nuclear genome if you're just mutating one of the genes in the mitochondria. So having the ability to go in and make gene mutations in mitochondrial genes is going to have a huge impact in better understanding this relationship between mitochondria and the rest of the cell. So with that, I'll just, my last slide is the promise, and that's really up to you. You'll have to really help us understand how the genome editing tools that we're all excited about can actually be brought to bear on some of these complex and important questions in genome biology in environmental health science. So with that, I will stop, and happy. Do I have time for questions? The timer is totally messed up right now.